422 million people worldwide are currently living with diabetes. And as you know, this is a condition that people get when their body can't control the amount of glucose that they're taking in. I'm going to be focusing on type 1 diabetes, which is where the body cannot produce enough insulin, which is the hormone that controls the amount of glucose in the body. And this can lead to a number of side effects like hyperglycemia, which is when there's too much glucose in the body, and that can affect your eyesight, your hunger levels, and so many other things. Currently, people are just using insulin, injecting the insulin into their body to control this disease. But the problem with that is that it's something that's constantly on their mind. Every second of every day, they're thinking about how much insulin am I going to inject today? How deep am I going to let the needle into my body? And so on and so forth. Not to mention the side effects that this medication has on the body. The list goes on. That's why I'm going to be looking into how we could use stem cells to cure type 1 diabetes. This kind of research has already been done before in the past, but I'm going to be looking into how we can solve the barrier of immune rejection. But before we go too deep into the science of it, I want to first understand what genes are expressed between stem cells and insulin-producing cells. That way we can see which genes we need to express in stem cells so that we can get them to produce the insulin that we need. For this, I'm going to be taking you through the whole process from how we can upload the dataset into BenchLink and then view which genes are connected to which amino acids, considering how much each of them is expressed. From there, we'll be able to understand what genes we need to introduce into these stem cells so that they can turn into the insulin-producing cells that we require. It has already been known that insulin gene, the INS gene, is already connected to insulin. So that's already a gene that we have off the checklist. I'm trying to understand what smaller genes are needed, not just for the insulin producing of these cells, but also for the longevity and for them to survive in the environment of the pancreas. Overall, my goal is for us to come up with a medication that can enable diabetic patients to live a life without having to think about insulin each and every day of their lives. By differentiating the stem cells into insulin-producing cells, we can transplant these into the body and have them regain that power of producing insulin for themselves without having to inject it into the body. This medication may have a high initial cost, but it's way cheaper than having to constantly purchase the insulin that they are currently using. So let's get right to it. So when you go to your Google search bar and type in NCBI gene, this is where it will bring you after you select gene. And in the caption, we'll select Homo sapiens human because we're going to be dealing with human genes. And to start off, we're going to first download the gene for insulin-producing cells. And from different research papers and different research that has been done in the past, we know that it's the INS gene that's related to insulin. So you can put that under the gene symbol and just search that up. And once you do, it will bring you up to this page where you can see the gene ID, the symbol, the gene name, the scientific name, just confirm that everything is correct. From there, you can click the gene ID, which should bring you to where you can download it. Yes. So this will just give you more information about the gene, what it's related to. You can go through this if you would like. It also gives you various expressions of the genes that could be useful. But yeah, from there we can just download the dataset, and I already have it downloaded, so we'll skip that part. You can repeat the same thing for stem cells, and we know that the OCT4 gene is also highly related with stem cells and their pluripotency factor. It's one of the pluripotency factors actually, and you can search that up as well, and it will take you through the same process, looking at the gene ID, the different informations about the gene. If it freezes, you can just refresh, and it will surely bring it up. So this one has quite a few variations, and try to pick the first one because the rest have different things correlated with them. But this is the one that we are looking into. So gene ID 5460. Once you click the gene ID, again, it will bring you into more information about the gene and what you need to know. And again, expression data, which is very, very crucial, so you can go through it during your own time to try and understand what proteins are involved, but we're going to get to that in our own way. So when you click download datasets, it's going to give it to you options. So you can choose the faster form, and then you can download it here, and it will be a compressed file, of course. So you can uncompress the file and convert the faster form to a CSV file so that you can easily upload it on BenchLink. So another thing that we know from our research about type 1 diabetes is that it's related to the HLA gene. So you can type in HLA under the gene symbol. So you can obtain a gene connected to diabetes, so that we can also see what genes are connected to diabetes and what genes are they lacking, so that we can be able to analyze those as well compared to the insulin producing ones and the stem cells. And of course, to take you through that same process, so you download the dataset, convert it to a CSV format, and then we can move on to Benchling. So once you've logged into your Benchling, you can come to the plus sign and click create, and you can go to DNA or RNA sequence and import from spreadsheet, since we have made it a CSV file, that's a spreadsheet, and you can select a folder where you're going to be keeping that information and the nucleotide, which is definitely DNA because we're dealing with DNA. And then from there, you would be able to choose the file and upload the different files that you've compressed and turned into CSV files. So I already have my datasets uploaded and ready to view. As you can see, this is the INS gene, ID 3630. And here we have our other gene that's related to diabetes, so gene ID 3123. And you can use these gene IDs to search them up if it's easier instead of going through the whole gene symbol process. And lastly, our gene ID 5460, which is related to the stem cells. So the next step is quite easy. What we're trying to do basically is analyze what amino acids are highly expressed in this genetical sequence, and then we can use another software which is called Protein Blast to understand what genes are connected to these amino acids, and then we can see what genes are being expressed in each data set and what genes are lacking, or what the differences in the genes are. So you can easily just select, you can highlight from the beginning of the sequence all the way to the bottom. It's quite a large data set. Once you do get to the bottom, you're going to want to click Analyze, and then you click Analyze as Translation, and that will bring you to all these different properties here, which you can see on the side. So once you've highlighted the whole of the data set, you can clearly see here that it's only GLU amino acid that's 100% expressed. So we can take note of that, and we shall later on go to the protein blast and see what genetical sequence is connected to that amino acid. So once again, when you go into your Google search bar and just type in NCBI Blast, this is where it should bring you to. And from here, we can click Protein Blast, since we're going to be dealing with amino acid sequences. And we can input that in here. So we have GLU. And from there are so many other fields that you can fill in, but we just want to leave them as the default answers in the field. And just click on Blast. And there it will take its time searching, so we can give that some time. So in the meantime, we want to continue on to our next genetical data set, and this is the one related to type 1 diabetes. So we can repeat the same process of highlighting the whole genetical sequence. Remember to highlight it all the way to the bottom. Once that's highlighted, we go back to Analyze and Analyze as Translation. And again, that gives you different amino acid frequencies. And this one's a bit more intricate since it's related to something a bit more complex. So we can again note down like the top 3 amino acid frequencies and what the amino acids are. And then we'll go back to the protein blast and see what genes are connected to that. So from this, we can see that GLY, SER, PRO, and VAL all have the same, amino acid frequency. So I've noted those all 4 down, and we're going to go to the protein blast and input. So for our first data set, we only put in a single amino acid, which was GLU, which was 100% expressed. But I think, as we all know, you can't have a protein with one amino acid, so there was really nothing. But for this next one, which we had GLY, SER, PRO, and VAL, we have a bunch of proteins and genes that are coming up. So once we look for the ones with top identities, which are these 3, and that's what we're going to take keen attention to and use in our next projects, etc. So that's what we needed. Now we're just going to repeat that for our final data sets, which I believe this was our 3123. So we're going to move to 3630, which is the last one, and I think, yeah, it's the last one. So we just need to again repeat the same steps, highlighting the whole data set from beginning to end. And once that's highlighted, again, we go to analyze as translation, and we can see the different frequencies. So remember, we're looking for top 3 or top 2 frequencies. Top 3, better, because you can't have 2 amino acids leading to a protein. So we can see ALA has 16.3, GLY has 14.3%, and PRO has 14.3. So you can already see the ones that are repeating. So we'll just take note of those. ALA at 16.3, GLY at 14.3, and PRO at 14.3 again. We can just get one more since we used 4 in the previous one. And right off the bat, I'm seeing SIR with 8.3. So we come back to our NCBI blast, and we're going to input those here. So ALA, GLY, PRO, and SIR. And we'll leave the rest to be filled in defaultly. Come to the bottom and blast. And it will take some time, so it will constantly keep refreshing and searching until it gets an answer. And just like that, we have our different proteins or genes that are related to this, and you can see this has 100% query cover. So again, you can take a screenshot or you can download this, which would be better. And from there, you have all the information that you need. And I'm so glad that you walked through this with me. And we can use this information to further analyze what we need to do with the stem cells that we have so that they can become beta cells, which are going to produce insulin in the body. So we have been able to see which genes are connected to producing insulin and what exactly we need to do with the stem cells that we have so as to cure this disease completely. From here, I'm going to be writing a review research paper to understand the different kind of research that has been done in this space, so we can understand not only the disease on a deeper level, but also what next we need to do with this information. So please stay tuned. Make sure to check out my Medium, because that's where I will be posting all my articles. You can also follow my LinkedIn, and all those are linked on my profile. Thank you.